Hi, and welcome back to DNA Unraveling the Code. I'm Dr. Kim Davenport, and today we're going to discuss how we extract DNA and genotype in the lab. We sampled our animals at the Beef Center, and so now I'm going to show you how we get from a tissue sample we collected in a TSU to DNA that you will use for genotyping. In the lab, what we do after we sample with a TSU is we take the tissue sample in the TSU and we put it in a solution of digestion buffer, which contains salt and detergent. Both of these will help break the cell membrane and release the DNA from the cells. We also add an enzyme called proteinase K. Proteinase K helps digest a lot of the proteins. And so we're gonna go from a large tissue sample like this to a solution full of lysed cells and free-floating DNA. After we digest our tissue, the solution looks like this, which is free-floating DNA and lysed cells. And to bring the DNA out of solution to retrieve it and use it for genotyping, we're going to add ethanol. Now we're adding ethanol to our tissue sample, which will bring our DNA out of solution. So you can see the DNA coming out of solution there as this white clump right here. Okay, so after we extract our DNA, we will prepare to load it onto these genotyping chips. Each one of these chips contain anywhere from 50,000 to 800,000 single nucleotide polymorphisms, or SNPs. And these SNPs represent genetic variation across the entire genome for lots of different species, including cattle, sheep, goats, dogs, horses, etc. When we're ready to load our DNA onto the chips, we load them into individual wells here. And I'm going to show you that piece right now. So you would take your DNA sample and load it into any of these individual wells. So after we load the genotyping chips for each sample, the chip will be placed into a genotyping instrument, which will calculate the genotypes for each animal and output the results. Not only can we genotype our animals, but we can actually sequence the entire collection of their DNA. We do this frequently in the lab. One way we do this is using a sequencer about the size of a stapler. And with the sequencer, we can insert a number of different, what we call flow cells, which is what contain the DNA for sequencing. There's a couple of different types of flow cells we can use, but really all of these accomplish the same goal, which is sequencing an animal's entire DNA sequence. Not just genotype the known genetic variation in an animal, but they can sequence an animal's entire collection of DNA, both known and unknown genetic variation, so that we can understand more about our animals' genomes and genetics and how that contributes to traits that we're interested in.